From here on, let me start the medical biotechnology session. Once again, it's a follow-up session of my earlier class. So the lecture which are based on online resources, particularly as you remember about NCBI and serial cloner and the software, you need to play with those things first. And then you generate your questions, and then you generate your troubles. And you encountered certain trouble in the meanwhile, and then that is the time that you can share those things with me, or you can share simply your experiences. I mean, you are not supposed to ask questions. You can share your experience कि मैंने उस जिनको किया, मैंने ये देखा, मुझे अच्छा लगा, मुझे नहीं अच्छा लगा, weird है, bored है, boring है, मैंने इसको enjoy किया, I felt differently. So whatever you want to speak about that thing, you can speak, okay? But let me just remind you about the last lecture quickly. I will go through certain things for you, and then you will be able to ask your questions with me, okay, in the meanwhile. So let me share my screen with you. You should be able to see my screen now. And if you remember that we were working that how to extract and get the sequence of a certain gene. Remember, the messenger RNA came from a portion of the genome which is called as a gene. And it is not only the protein coding region. It contains the 5' UTR region, it contains the protein coding region, and then it contains the 3' UTR region. No introns. There was no introns. From where we get the sequence of the introns, that's a different question altogether. I will show you how. Don't worry, nothing to worry about that as well. But since the sequence we were working in our last class, that is about the sequence of insulin. So whatever you were looking at that time, that was only the protein coding sequence, which is called CDS, 5' UTR, and then the 3' UTR, together with the CDS. And let me just repeat that activity quickly. You need to go to NCBI, go to the gene if you want to get the sequence of gene. That does not mean NCBI is only meant for this purpose. NCBI can do a lot of things, lot of things, just nothing which I taught you. And I want you people to please play with the other components and try to read the other things as well in the meanwhile. Do not depend on anybody, I mean, including your instructor as well. Depend on you, that it is your own specific personal development. You need to add things in your CV. At the end of the day, I mean, at the end of the lecture, you can add these qualities in your CV as well, that you know these portals and these software. Okay, and then we were asked to, somebody recommended insulin gene, and the symbol is, I believe, INS. And then we go directly the insulin gene organized by the NCBI people. I explained you about the things on the first page, different important components, about the papers, and then I ask you to stop at the reference seek, NCBI reference seek. And then I explain you about the transcripts. And then we selected one particular transcript, this one, to study further. I explained you about this page, what are features, how these features can be understood, what are the information we can get out of this page, about the protein sequence of the gene we are talking about the gene sequence we are talking about, and we have made a file out of it as well. We just selected that sequence, we copy it, And then we created a raw material, a raw text file to put that sequence as a raw sequence here. That was done already. Then I introduce the serial cloner software. I mean, I'm not supposed to teach you just one software. I'm repeatedly telling you, very important. This is not one and only software. This is just one of the free software. Okay, you might be able to end up with a better software with more options and with more more user-friendly options. Then I pasted the sequence here, and I teach you how to put the features, how to label the DNA sequence, to add customized information at different positions, and how to save that file. Click on the file, click on the save, and then you can type the name of your choice, and then you can save that file. So last time we ended up with this file. I named it like sample insulin at that time. I told you should be very specific, and you should put as much information in naming the file, which can help you to go back and to find and to make sense more easily about a particular file in your computer. Because when you choose to stay in one particular area of research, whatever you are generating in the meanwhile, these are your powers. These are your power tools. This is a very simple file, but I can make it very complicated over the period of time. I can put more features, more information, and more background data as well in that file. Last time I did not share much of the functions available in these softwares. I'm just sharing whatever is like as a novice or a starter, you should know. But that thing should not stop you to try the other options. Okay, you are supposed to play with these things. You're supposed to try and see what more you can do. Okay, just let me share one more thing. We studied about the sequence map. I ask you to check this box. Do not show restriction enzyme RE sites so that it will look easier. It contains both the strands of DNA as well as the 3 translational frames. I explained the translation frames as well. You can define the number of nucleotides per line as well. By default, it is 100 nucleotides per line. You can change it to 60, 40, 50, whatever you want to. You can change a little bit the size as well, according to your understanding. And you can select which of the frame you want to study. Last time we studied that the frame number 3 is the translational frame of the insulin gene. If you remember, because it starts with the methionine, no stop codon during the consensus cDNA sequence, and the first stop came where TAG, the stop codon. Okay, I am just putting a smaller size. So that is how you can see the map of whatever you have created. Different colors, you have given those colors, you have given these names. If you remember, 5' UTR, start codon, consensus cDNA sequence, stop codon, 3' UTR, brilliant. Now what more we can see, we can use this thing. If you found a sequence of an insulin, or if you want to introduce a mutation into a particular gene, since I'm using this one gene, insulin, you can try with the multiple genes. That will be your assignment as well over the weekend. If I want to make changes in this sequence, or if I want to create mutation in this sequence, you know, if I created a mutation of one base pair, that can change the amino acid as well. That can also change the frame of the sequence as well. If I delete two base pairs, that will change the amino acids. Answer is yes. But that will also change the frame of the sequence as well. Frame will be shifted as well. If I change the 3 base pair codon, it will remove one base pair. Sorry, it will remove one amino acid. It will remove one amino acid, but it will not going to affect the translation frame. Because if one codon is gone, that amino acid will be deleted, but the frame will be intact. You can see all those theoretical understandings which you develop over the period of time when you study molecular biology and other things about mutations. You can actually see those things with your own eyes, that what will happen if I add a base pair, what will happen if I add 2 base pairs, what will happen if I delete 3 base pairs or add 3 base pair or 5 base pair. So you can actually see the difference after the mutation. So let's quickly just try to add a mutation somewhere in that sequence. How about that place? Any place we can choose, it's our choice. For an easier understanding, I am using something right after the start codon. Okay, my mouse cursor is there. This is the position number. I am highlighting it. It's very small, but in the video you will be able to see it clearly. The video is HD. So I am highlighting a base pair called G. I don't know is the first member of the codon, the second member of the codon, or the third member. I don't know anything. I just want to change that base pair. And then I want to see what happens with the protein sequence. This by changing, this is the base pair number 76. You can see here. I have selected one base pair. And the tool is telling me is a base pair number 76. So what we should do is our own choice for learning. क्या करें? Base delete कर दें, remove कर दें, replace कर दें. Suggest me in the text box.
Replace कर दें, sir.
Who is speaking? Usama. क्या करें? Usman क्या करें? Eamon, Anisha. फरहान देखो कितनी मजे की बात है, जो मर्जी करे इसके साथ. Tell me, we will going to act like gods. हाँ, क्या करें?
Replace कर दें.
Replace कर दें. Okay. First suggestion, I can see replace कर दें. I have more suggestions. Sir, delete it. Other one, replace it, delete it. 2 replace, 2 deletes. Even Steven. I just want one more casting vote. Delete करें, replace करें, क्योंकि फिर बाकी आप खुद करेंगे. Delete it. Okay, I got the vote. So let's delete the base pair number 76. किधर गया? The base pair number 76 called is G, is right after the start codon. So if you want to make changes in this sequence, you need to see certain things. Number one, whether the sequence is locked or not. If the sequence is locked, and you should keep your sequence locked after working. So कोई बच्चा जो है वो शरारत में आपका sequence बदलना दे, that can haunt your research a lot. Primer sequences and other sequences. If it is locked, you can't make any changes. So first you need to uncheck the box. So it will allow you to make changes at the place of your choice. And what is the place of your choice is the 76 base pair G, which is highlighted. Actually, it is highlighted. So you know the file name. The file name is sampleinsulin.txt. I will going to delete that gene. You can delete that gene simply using the backspace. Good enough. Okay, backspace. Now the G is gone. Since the one base pair is gone down, that will change the length of the features as well. That's very important. So you will have to adjust the features as well if you make changes in the file, because the features are saved based on the start of the feature, start number of the feature, from where it is starting, 20 base pair, 25 base pair, or where it is ended. So if you delete one base pair, the numbers are shifted. You should remember that thing. So once now we have a new sequence. Let's save that sequence with a different name. Save as sample insulin 76 base pair del deleted. Okay, this is a mutation, new name. Save. So now we have two sequences. We have two files. One file with the sample insulin sequence deleted 76 base pair, which is G, and the other file is a wild type. And we can see with our own eyes what is the difference between these two files. Okay, so let's browse that sequence, 76 del sequence. I will go to the sequence map. I'm sure you understand and remember the things. I am asking the software, do not show me the restriction enzyme sites. I'm not interested in frame 1 and frame 2. These are not the translation frame anymore. Let's see. It will start from where? M. The 76th base pair should be, this is the base pair number 70. Okay, remember 71, 72, 73, and here something bad happens here. I don't know that was bad or not. Let's see. Now remember the sequence translational frame, methionine, then A, then L, W, M, and then there is a P out there. A P is here. And then S, then C, then P, then C. Just remember that thing. This is supposed to be the translational frame. That means no stop codon should come before the wild type stop codon. The wild type stop codon is here. It is shifted one base pair, but it's here. Now you can see, just go down to the amino acid sequence. Go down, go down, go down. Oh my God, look at that here. You can see one start codon appeared. That was not there in the wild type sequence. What that means? When the protein synthesis system start making the protein, this start translating this gene. Because of one base pair difference here, it will shift the frame and the protein translation will stop abruptly here. This is called truncation. This is called truncation because of frame shifted mutation. And that mutation which you just have created is called as the frame shifted mutation. Just compare that sequence with the wild type. Where was the wild type sequence? Here is the wild type sequence. Let's go to the sequence map of the wild type sequence. Ask, do not show restriction enzyme. Frame 1 is gone. So I am on the wild type sequence now. And I want you to compare these two sequences with your own eyes. That what is the difference between the two sequences? Let's go for the first line. M is fine. M is here in the wild type. The above is the wild type sequence. M is here. Yes. M is here too. Yes. MAL. MAL. WM. WM. And then there is a P. Then there is a P here as well. Oh, I'm sorry. I'm using both the deleted sequences. I'm closing one of them. Sorry. I should use this one. This is the sample insulin, not this one as well. I have to open the wild type sequence. It was closed, sorry. Here we go. Here we go. The wild type sequence. Show me the wild type sequence. This is the wild type sequence. Let me go to the sequence map of the wild type sequence. Do not show me the restriction enzyme sites. Frame 1 is gone, frame 2 is gone, and let me help putting these files here. Wild type sequence, and this is the sequence of the mutated file. This is the sequence map of the mutated file. Okay, here we go. Above one, is the file for the wild type. Here is the file. Actually, when I shift the files, it changes the sequence. Just try to use your memory to remember. This is the file for the wild type. MALWM, and then there is RLLP. And after that, there is no stop codon until the wild type stop codon came. Let's move to the deleted file. This one. It starts with the M, goes to the ALWM, and then the sequence changed. The sequence of product amino acid will be changed. P, S, C, P, C, W. It's different. This sequence is different than the wild type sequence, which is MALWMRLLP, and there is no stop codon here. But in the wild type sequence, when you change one base pair, it actually changed all the amino acids after that, and it also incorporated a stop codon there as well. So not only the proteins or polypeptide chain sequence is changed, but also it is encountered with an unwanted stop codon, and it will not end up in any protein or any functional protein. One base pair change. I request you people to change one base pair, then two base pair at one location, then other location, add one base pair, add two base pair, add three base pair at one location and the other location, and see what happens. Okay, you will find many interesting things out of it. Do not do this for any course. Do it for yourself to see that if you're able to find some more interesting thing out of it or not. For example, if you change the third base pair of any codon, if you change the third base pair of, it arranged in codons for you. If you change the third base pair of any codon, you might not see any change in the amino acid. The next amino acid might be the same. The amino acid sequence will stay same. If accidentally the mutation occur in the third base pair of every codon, it do not make any changes in the protein sequence. Mutation hai, but not causing any trouble. Silent mutation. So you suddenly came to know with your own eyes that if nature install that type of mutations in my sequence, how it will be ended in the protein product form. Okay. Okay, before I stop, there are a lot of things I should tell you. I mean, khatam nahi hoengi cheezein, aa hi jayengi. So let me share one more thing with you before I stop that thing. I'm closing everything for you. Now we have two sequences, if you remember. The one, sample insulin, the other one with the 76 base pair deletion. If you have two sequences, you can compare them. And you know how you do this? You can use NCBI BLAST for that purpose. You are asking somebody else to do the job for you. And then after that, you might have a company with some of your seniors and more learned students. They put some sequences in the computer, and then they click a button called BLAST, and then they wait, and then they get the sequence. Then they start telling you something very, intellectually supreme things, like they are stars of science. They are nothing. These things are nothing. You can do these things manually with your software. Now, for example, this is the same situation. I have two sequences. One is wild type, other is mutated. I can compare them. I can compare them to see. I think मुझे second session लगाना पड़ेगा. मैंने सोचा था नहीं करेंगे. Anyway, so let's see. Let me how we can compare them. So the software can help us to do a lot of things virtually, and it can also help us to align two sequences. You need to go to the function and then align two sequences. It has a lot of options, very easy. Whatever I'm telling you few minutes earlier, you can see all those things in one go, in one screen. Ask me to select the sequence number 1, I am putting the sequence called sample insulin. And remember, this is a wild type sequence. The wild type sequence. The second sequence, I am putting the 76 del, the one with the deleted base pair. हमने खुद ही किया. And then what are the frames you want to see? You know, we want to see the frame number 3. If you remember, if you don't remember, forget it. If you don't want to see the amino acid sequence, still fine. You can pick and choose whatever you want. Are you little bit excited? You want me to click on the button to see what happens? If there is something kuch hota hai out there in your heart and in your mind, that means things are going nice and fine with you. You're getting something. I mean, that's the beauty of knowledge. It should create a little vibrations in you. Okay, dekhte hain kya hoga, what will happen. So I am asking and clicking the button called local align. I'm not asking the help of anything. I don't need an internet. I don't need anything. I just need my sequences, and then I have that software. So let's click on the local align, and local align will give me the comparison of these two sequences. In order to read these, you know, अब लगेगा ना किसी को pressure डालना ना अब आपने किसी को अपने साबित करना है कि आप एक अजीमुशान scientist है. You can use these type of things. Oh my God, क्या मजे की बात लगी. You know what is sequence 1? You should know what is sequence 1. Sequence 1 is this one, which is called the wild type. What is sequence 2? Sequence 2 is this one, the mutated one. Everything is same until the 60 base pair. Yes, no changes. Everything is same here. Or did you find anything weird in the comparison? There should be something weird. You know, कहाँ पर होना चाहिए weird? 76 base pair पे. Can you see a weird situation here at this GCGC? This is the sequence CGC. And what is the sequence here? C, nothing, C. So a base pair is not here. So when the alignment is done, it is telling us that the sequence number 2 has a missing base pair here. You can see the parallel lines. There is no parallel line here. If it is deleted, it will be appeared in a different way. If it is substituted, it will appear in a different way. And you can see it is showing us, okay, at that base pair, that there exists a difference. You can actually see the frames as well. This is the frame for the sequence 1. This is the frame for the sequence 2. जो मैं अभी इतनी मुश्किल से different windows लगा के आपको दिखा रहा था. Check the frame. ALWMRLLP. ALWMPSCP. Which one of them is the wild type? Not this one. This is a wild type. Or this one the wild type. कौन सा इनमें the wild type है? ऊपर वाला है कि नीचे वाला? You can answer me in the text or you can speak. ALWPMR. This is a wild type or this is the wild type?
ऊपर वाला.
ऊपर वाला wild type.
ऊपर वाला सर.
ऊपर वाला wild type है, नीचे वाले हम सारे mutant हैं. Okay.
Sir,
good.
sequence 1 is the wild type.
Shabash. Thank you. So there should be no stop codon in the upper amino acid sequence until unless the wild type stop codon appears. But because of the shifting of the translational frame, you can see a stop codon here. So no protein translation will be there after that. Okay, so that is how you can play with that software in a different way. And you should try all these different things, and you should enjoy and try to exercise and practice. There is no shortcut. मैंने बहुत कोशिश की है क्योंकि मैं खुद को दुनिया का सबसे ज़्यादा lazy इंसान समझता हूँ. I wanted to do things with the least possible efforts. मेरा दिल नहीं करता काम करने का, hard work करने का. So whenever a work is given to me, I try to find a way how not to work. That's the first thing. Second, I try somebody else to do my work. But if unfortunately I have to do that thing, I'll try a way to have that thing done with the least possible effort. But I came to a conclusion that if you really want to develop an expertise in one particular area, there is no shortcut. You just need to spend hours and hours in that thing. Okay, my student says that we are same like you. Thank you very much. At least we are common in this thing. So the point is that, which I'm telling you, when you see a sketch from an expert artist, you can feel the amazement. Wow! But that amazement came out of the several hundred hours of effort that person had spent out of his life. Like कुछ भी कर रहा है वो draw कर रहा है, फारिग बैठा है वो draw कर रहा है. He's actually educating himself or herself to draw these things in a better way. When you came to know about a new knowledge, like a new software or a new area, it is only you who can educate yourself better than the others. That is the real race. लोगों को लगता है कि मैंने अच्छी जगह से पढ़ा है, मेरा टीचर अच्छा है, मुझे अच्छे से पढ़ाया गया है, so that might be a primary factor. That is a secondary factor, not the primary factor. The primary factor is that how you can educate yourself with your own free will and wish. So I request you to please try as many things as possible out of these things, because we don't have enough time, and this is not the class like to spend all the time in this area, but I want to start. It's like a kick start in a new area to start developing more knowledge. For example, I wanted, so for example, last time I asked you about the consensus CDNA sequence, and I told you that the NCBI has a separate website for that thing. Do not wait somebody else to take you there. Please, please. Okay, एक बात पहले मान लें, कोई हमारी मदद नहीं करने वाला, कोई भी, मतलब कोई किसी की हेल्प नहीं करता. ये बहुत, I'm sorry to tell you this brutal truth. So when you heard something like in the last class, there is a separate website for the consensus CDNA sequence. Many of us already started believing वहां जाने का फायदा कोई नहीं है, मुझे वो आता नहीं है. I might not be able to, get things out of that website. Some of us might be thinking in a different way. वहां चल के देखते हैं वहां पर क्या होगा, try करके देखते हैं क्या होगा. Let's see. So just be like that one, okay? So I think I believe that we need to go and enter into the next second session, because I want to show that website particularly designed for the consensus CDNA sequences, the most easier way to get the consensus CDNA sequence of a particular gene, how you can get it, what are the variables out there, and then I will move to your question answers and comments. Okay, share your comments if you don't have any question. Share your experience कि मैं ऐसे फील कर रहा था जब मैंने किया, जब मैंने ट्राई किया तो मुझे शुरू में ऐसे लगा, बाद में ऐसे लगा. That is very important as well. Okay, last time I used that term a lot, like consensus CDNA sequence, and I told you that most of the people who are working in expression, cloning, and these type of things, they are mostly interested in looking at the genes, looking at that part of the gene which is coding a particular protein, and that part is called as the consensus CDNA sequence. And you can find that term almost in every other page to describe a gene on NCBI. Whenever NCBI is giving you the information, it will also give you a supportive information about the consensus CDNA sequence. There are many ways to go there. Whenever you find that information, for example, I'm browsing this page for you, and in the description, somehow, somewhere you will see this CCDS, the consensus CDNA sequence. You can also find that page and then manually input the information of insulin or use the code. I will show you where the code is. So if you click on this page, I am clicking it, it will take you to the consensus CDNA sequence page managed by the NCBI as well, but this is a different thing. particularly working with the CCDS sequences and database. We don't need to search anything here because we came directly from NCBI by clicking a link. So it will take us to the place where we want to. You know, the species Homo sapiens, because we were working on the Homo sapiens genes. This is the ID, this is the chromosome number, and this is the gene name, INS, and blah blah things. So you have the information. You will see certain other buttons here, and I request you people to please check and click and see where that button will take you. Actually, this button will take you to additional information about that gene present on other places organized by the NCBI and other portals. Okay, so when you go down and browse that page, consensus cDNA sequence page, it will take you to the sequence, nucleotide sequence of insulin. Certain notations you should know. This is the consensus cDNA sequence. No untranslated part should be here, first thing. So it should start from where? It should start with the start codon. Can you see ATG? If you place your mouse cursor there, it will tell you the name of the amino acid as well. It should end where? At the stop codon, TAG, nothing else. This is the same thing. If you click on the feature called CDS in the NCBI page, if I click here, CDS, it will take me to the same sequence here to here. But that sequence is cropped out and pasted here in a different way. You can see two colors, the black one and blue one. That indicates two exons. If there are three exons, the first exon will be in the black color, the next one in the blue, the third one in the black, the fourth one in the blue, the fifth one in the black, and that is how it will tell you. You will easily understand from where the first exon starts and from where the first exon ended. For example, it starts from here and ends here, the first exon. And also, if you select a certain sequence, for example, I am selecting that sequence, you will see a certain sequence will be, a protein sequence will be selected as well. If you click on a certain codon, CAC, it will tell you actually it will encodes H. If you click on another codon here, it will tell you you are talking about this place. If you click here, sorry, if you click here, it will tell you that you are talking about that place. You can actually pinpoint that which codon is incorporating which amino acid. One more thing you need to know, the splice junction. This is important. When two exons merge together, you know, spliceosomes karte hain ye kaam. They cropped out the introns between the exons, merge them together. So the amino acid which is joining the two is referring as a splice junction. Here, it's usually colored as red. हम अच्छे हैं ना, lucky हैं कि हमारे पास दो exons हैं, for multiple exons भी हो सकते हैं. Now I can use that information to make my file more beautiful. Like I am selecting, oh my god, I am selecting the first exon, it stops here. I am copying that sequence, only the first exon. I will go to my file on the serial cloner. I'll open the file, wild type file, which is sample insulin. This one is the wild type file we are creating. And I can find Control F here, and you can find that sequence, Control V, putting that sequence here. It will tell you that you have selected 187 base pairs. It starts from 60. It ends at 246 base pair. And this is, now you know, this is the exon number 1. And you can add a feature as well to make your file more special that this is exon number 1, or you can change the color as well if you like. Go back and the feature is edit exon number 1. And then similarly you can add more features. So jahan se, from wherever you get certain information, you can implant all those information in your local file, which you can use later on to write your thesis and to answer certain questions and to have a customized file of your own choice. If somebody is working on a one particular codon or the mutation present in one particular place, that person can highlight that region as well. So that is how you can use this page as well. You can directly search that page, CCDS. Okay, CCDS report. You can directly search that page. It will take you here, but here you need to search that sequence by yourself. There are many ways you can search it. You can search it using different means, using consensus CDS ID. If you know that ID is there, I can tell you that ID is there on this page. थोड़ी सी अकल जुड़ाएंगे तो you will get that ID 7729.1. This is the consensus CDS ID. You can use that ID. You can use the gene ID if you know. Gene symbol, INS. INS is the gene symbol. Nucleotide ID. If you remember, we have a nucleotide ID. Where? The nucleotide ID, if you remember the last lecture, NM. This one. Particularly for that gene. We can use this one. So you can use any of that option to search the product. An outcome you can search and you can go and it will take you to the place. ये वही ले जाएगा हमें जहाँ पर हमें. If you are putting the wrong information, you will end it up at the wrong place. You need to be very, very accurate in finding the sequence of the genes. Whatever we are studying.
Yes.
जी हमजा.
Sir, I have a question.
जी जी.
Sir, मैंने पूछना था कि एक जमाने में हमने पढ़ा था about codon optimization के plants में different codons होंगे for the same amino acids and whatnot. तो हम इस software को इस्तेमाल कर सकते हैं वो codon optimization करने के लिए?
सॉफ्टवेयर को इस्तेमाल नहीं कर सकते. सॉफ्टवेयर को एज ए राइटिंग टूल के तौर पर इस्तेमाल कर सकते हैं. If you are clear about the codon optimization, that is fine. For example, मैंने इसको ऊपर बात की हुई है किसी लेक्चर में. Remember मैंने इस पे बात की हुई, मुझे याद नहीं आ रहा. हाँ
बिल्कुल वो याद है.
जी 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 जी. सो अगर आप एक ह्यूमन जीन को, मिसाल के तौर पर ये इंसुलिन का जीन है, ये आपके सामने है. अगर आप इसी ह्यूमन जीन को किसी बैक्टीरिया में क्लोन कर देंगे एज इट इज, बैक्टीरिया विल नॉट बी एबल टू प्रोड्यूस द प्रोटीन एफिशिएंटली बिकॉज बैक्टीरिया सेम अमीनो एसिड्स को इंस्टॉल करने के लिए डिफरेंट कोडॉन्स को प्रेफर करता है. सो वी नीड टू कन्वर्ट द सेम सीक्वेंस. वी नीड टू मेक चेंजेस which is called codon optimization. So it should produce the same amino acids. So it should produce the same protein in bacteria with more efficiency. So if we know what are the changes we want to make, we know we need to change this G into C. We need to make changes in this C into T. If you know the changes, you can manually edit those changes in that gene to optimize it to be used in bacteria. The answer is yes, but that might be tricky. That is sensitive work. Any mistake might end up in a wrong experiment. So better to use online resources, like put the sequence, human sequence, into the portal in the Google. There will be certain sites for that purpose. Ask that site to convert that sequence or optimize that sequence for bacterial expression. It will give you another sequence. Copy that sequence and come back to your serial cloner and save it. So once again, now the new sequence will produce the same protein, but efficiently for bacterial system. लोग ये करते हैं क्योंकि कभी कभार लोग फंगस में एक्सप्रेस करते हैं प्रोटीन, इंसेक्ट सेल्स को इस्तेमाल करते हैं. तो दे हैव टू ऑप्टिमाइज द सीक्वेंस अकॉर्डिंगली. और ये बहुत इंपॉर्टेंट कॉन्सेप्ट है. मैंने इस पे पहले लेक्चर पे बात की. शाबाश जी. वो बोर तो नहीं हो रहे? सो तो नहीं गए?
सर
जी जी
सर
आपने वो अभी उसमें सीक्वेंस में रेड कलर में शो किया था स्प्लाइसिंग कोडोन जो था वो रेड कलर का था मेरे ख्याल से, बी जो था जो अभी इसमें सीक्वेंस में हम शो किया था सर, वो क्या चीज डिटरमिन करती है कि कौन सा जो कोडॉन होगा या अमाइनो एसिड है वो स्प्लाइसिंग होगा, या हर दफा सेम होता है वो दो एक्सॉन्स के दरमियान?
नहीं नहीं, ऐसा नहीं है, ऐसा नहीं है, ऐसा नहीं है। एक्चुअली हमें कैसे पता लगता है कि ये एक्सोन है, यहाँ पर एक्सोन खत्म हो गया और यहाँ से एंट्रॉन शुरू हो गया? दिस इज योर क्वेश्चन, एम आई राइट मुस्तफा?
यस सर।
हाँ, दिस इज योर एग्जैक्ट क्वेश्चन। कि एक जीनोम के ऊपर एक सीक्वेंस है, हमें कैसे पता लगेगा यहाँ पर इंट्रॉन खत्म, यहाँ पर एक्सोन स्टार्ट? सो देयर आर सर्टेन सिंबल्स, साइंस, निशानियां होती हैं, निशानियां जीन सीक्वेंसेस के ऊपर निशानियां होती हैं which can help the spliceosome machinery to get a sense that okay, now this is the termination of an exon and this is like the start of intron. So they use those signs and symbols. छोटे छोटे बेस पेयर्स होते हैं, एक एक दो दो तीन तीन जिनके साइंस होते हैं. You should see those sequences as well. Okay.
सर वो मैंने पूछना था कि ये जो फास्टा फॉर्मेट और cDNA इसमें क्या फर्क है?
फास्टर फॉर्मेट और बात होती है, cDNA और बात होती है. कंपैरिजन नहीं कर सकते. जब हम न्यूक्लियोटाइड सीक्वेंसेस को लिखते हैं, न्यूक्लियोटाइड सीक्वेंसेस को जब हम लिखते हैं तो उसको लिखने के डिफरेंट फॉर्मेट्स हैं. फास्टा फॉर्मेट उनमें से एक है. लिखने का मतलब ये है कि हम नंबर लिखेंगे कि नहीं लिखेंगे, हम स्टार्ट का सिंबल क्या डालेंगे, हम खत्म होने का, टर्मिनेशन का, एंड सीक्वेंस का सिंबल क्या डालेंगे, हम कितने बेस पेयर्स का गैप डालेंगे, कितने स्पेसेस डालेंगे दरमियान में, 10 बेस पेयर्स के सेट्स में डालेंगे या कॉन्टिन्यूसली डालेंगे. दैट इज कॉल्ड एज अ फॉर्मेट. तो फास्टा फॉर्मेट इज वन ऑफ द फॉर्मेट. जीन बैंक इज अनदर फॉर्मेट. cDNA सीक्वेंस जो है ये और है, जो मैंने बताया आपको कि प्रोटीन कोडिंग पार्ट ऑफ द जीन जो है वो cDNA सीक्वेंस होता है. देयर आर मल्टीपल फॉर्मेट्स टू राइट द सीक्वेंसेस, न्यूक्लियोटाइड सीक्वेंसेस इन डिफरेंट वेज. और ये मल्टीपल फॉर्मेट इसलिए हैं because people are developing bioinformatics softwares and different softwares are optimized in using different types of formats. So एक फॉर्मेट है जो कहता है, आपसे मुझे एक सॉफ्टवेयर आप इस्तेमाल कर रहे हैं, वो कहता है मुझे फास्टर फॉर्मेट में सीक्वेंस दो. So you need to give them the sequence or submit or input the sequence in a faster format. If it is asking you to submit the sequence in another format, you need to convert that faster format into another format. So portals जो है वो तमाम options जो है वो आप सामने रखते हैं. जी कतीश, sir, why was 3 more than 1 frame for each sequence on the software? Why was there more than 1 frame? Because one sequence of nucleotide can generate 3 frames. It depends from which base pair you start translating. If you are starting the translation from a particular base pair, the next 3 base pair will become like a codon, and that codon will incorporate a amino acid. If you start translating, if the system start translating from the second base pair, the next 3 base pair will be acting as a codon, and that sequence will be different from the first one. So every one single nucleotide sequence can produce 3 translational frame. So the software is giving you all these 3 options. Now it's on you. अब ये आपके ऊपर है that you should be able enough to figure out that out of these 3 frames, which one of them is the true frame or the true translational frame. And I shared that thing, that which one of them will be the true one. Boy. So next question is that why the third nucleotide in a codon do not result in a change? Why not first or second one? Actually third, ये evolution है. This is an evolution. बस हमारे पास एक अमीनो एसिड को इंस्टॉल करने के लिए 3 कोडोन भी हो सकते हैं, 4 भी हो सकते हैं, 5, 6, 7, 8 भी हो सकते हैं. जब आप उन 5 और 6 अमीनो एसिड को स्टडी करेंगे, you came to know उसमें पहले 2 सेम होंगे. Usually the last one is changed. So there is no logic other than the evolution, that the evolution actually selected multiple codons to install one amino acid. And if you change the first one, it is highly likely, 100% chance that the amino acid will be changed. But when you change the third one, usually it ended up in the same amino acid. इसका मतलब ये नहीं है कि वो बदलेगा नहीं. Codon will be changed, but the new codon will install the same amino acid.
Allah Hafiz,
Hafiz,
sir.
Allah Hafiz, Allah Hafiz.
Thank you, sir. Hafiz.
Allah Hafiz.
Bye bye, Farhan.